This lecture will discuss Mendelian genetics. So Gregor Mendel was a monk that studied inheritance using pea plants. He actually wanted to know why certain plants had certain characteristics, and he would look at their colors and their seeds. So we're going to talk about the seeds first. And what he did, he crossed plants that had either a round, smooth seed or they had wrinkled seeds. And he did this by actually brushing some of the pollen off one plant and then cross-pollinating with that brush and putting the pollen on the other plant. And what he did, if you look at this, we have a round seed and a wrinkled seed. And this was called the parent generation. When he crossed them, he ended up using a homozygous recessive, which is the wrinkled seed. Okay, so you see the homozygous means that the alleles are the same, and we're going to talk about that a little bit. And recessive being, opposite of dominant, and we'll see that a little bit more later also. And you see we represent these by using the lowercase letters. And then he also crossed it with the round seed, and this was a purebred, a homozygous dominant, so this is also purebred because the alleles are the same. So homozygous dominant, we see two capital alleles, and we'd get one allele from mom, one allele from dad. And when he crossed the parent generation, he ended up with the first filial generation, filial actually meaning son. And the results were a round seed, but what we came to later find out was that it was actually heterozygous because when he crossed the F1 generation to itself, so he took these offspring and actually cross-pollinated them, he ended up with the second filial generation and ended up with 3/4 round seeds, or 75% having round seeds, and 25% having wrinkled seeds. So somewhere it was hidden, and then it came out again. So if we cross the F1 generation and a homozygous recessive, homozygous again having the same alleles, recessive being the lowercase, the non-dominant allele, we can see that we're going to get 50% of the smooth seeds and 50% of the wrinkled seeds. But here, if we were to look at the alleles, which you wouldn't see, you would actually get heterozygous individuals. Then if we cross two heterozygous parents, we would end up having a multiple combination. We would end up with 25% being homozygous dominant and 50% being heterozygous, and then 25% being homozygous recessive. So then that appearance or phenotype reappeared when he crossed two heterozygous individuals. So this led Mendel to come up with some rules of heredity. And he wasn't using the terms yet that we use now, but started describing them, which were later termed. So the first one, he said that there were factors. Now we call those genes that are passed down from one generation to the next. So if we go back and we think about the parent generation, he had a round seed and a wrinkled seed. And then somehow it disappeared in the first filial generation, only got round seeds. That wrinkled disappeared. And then when he crossed the first filial generation, it reappeared, the wrinkled seeds. He also said that every gene has two variations. There's a dominant and a recessive, and these are called alleles. And then every organism is going to inherit two alleles, one from each parent for a trait. So then that's where we get this term heterozygous or homozygous. Heterozygous being hybrid, having two different alleles where the dominant is going to show through and you won't see the recessive, or they're homozygous, which can be homozygous dominant and having the dominant trait, or homozygous recessive, which means they have both alleles for recessive. And we would only see the recessive if it's homozygous recessive. And this is where the law of dominance came in. It said in heterozygous individuals, only the dominant allele is going to be expressed, hiding that recessive. So we wouldn't see that it's there until offspring were produced. and the law of segregation. Alleles are going to separate during meiosis, so gametes are only going to carry one allele for a trait. And due to independent assortment, and we get variation because of sexual reproduction and meiosis, we're going to get different combinations of those alleles.